Hey everybody, welcome back to General Biochemistry. We're still in Leninger, Principles of Biochemistry. We're going to be using this textbook the whole time, but occasionally I may pull from a paper or from another textbook just because I think that it kind of gives a little bit better of an overview of the topic that we're covering. Just a heads up. We are on chapter 5, and this is protein function. If you have not finished watching chapter 4 on protein structure, stop this video, go to chapter 4. You really need a good understanding of protein structure because we're going to be mentioning structure a lot in this presentation. So if you don't have a good idea of your secondary structures and the amino acids from chapter 3, then you really need to make sure that you're good on that before you come to this chapter. We're actually only covering the first 2 sections. We're covering section 5.1 in its entirety, pretty much, and section 5.2, just a kind of a selection from that. But don't worry, there's plenty of content for us to chew on. Now, I usually kind of say, all right, let's keep going. But one more thing. For this chapter, we're going to go over a few case studies to look at protein function and how it's related to structure. You will need to know the information and how these kind of protein systems work, but you'll also need to be able to interrogate a new system that I introduce. And so make sure that you're paying attention to the types of questions that you'll need to ask and answer when it comes to digging up new information about a protein or how it interacts with another protein or ligand. So that's just a little heads up for what you'll see on quizzes and exams. Here we go. As always, here's the list of key principles that you'll be able to talk through, give examples of, etc. at the end of this chapter. Always make sure that you revisit this page and know that, okay, yeah, I can see that. Yeah, we did talk about that. I understand that. And you can expand on each of these points a little bit. So overall, when we characterize protein interactions, we've really got 2 types. We've got a protein that's acting as a reaction catalyst or an enzyme, and we'll get to enzymes in chapter 6. Enzymes are going to alter the chemical configuration or the composition of a molecule that's bound to it. So there's chemistry happening. Something binds, and when it leaves, it is not the same. The other type of interaction that we're going to be talking about in this chapter is when the chemical configuration, composition, they're unchanged. We just have a protein binding some kind of molecule, and then maybe that makes some kind of a change in the cell, or maybe that molecule is transported somewhere else and then released. But we're not doing any chemistry. We're going to start with oxygen binding proteins. Oxygen binds to heme groups. If you remember from chapter 2 when we were talking about intermolecular forces, we also touched on nonpolar gases. Gases do not like to be in solution. They're very terrible. They're poorly soluble. And diffusion through tissues is really ineffective over large distances. So if you have to get oxygen all the way from your brain down to your toes, that's not going to happen just by diffusion alone. Not to mention, within the body, you have all kinds of other molecules that oxygen can interact with, particularly metals. And metals have a strong tendency to bind oxygen. So that oxygen is not going to get to all the places it needs to go if it's just left to its own devices to diffuse through your body and get to the tissues that it needs to get to. Enter the heme prosthetic group. Heme is present in both myoglobin and hemoglobin. We're going to talk about what those 2 proteins are. Both of these proteins have a protoporphyrin, which is the actual heme group. Heme is a protoporphyrin. And in the center of that, you have an iron 2 atom. And that iron is coordinated in with six different atoms that are all from the protein itself. So the protein has all of these amino acids that form this site that the hemoglobin sits in nicely. So like I said, there's 6 coordination points. You can only really see 4 in the first 2 figures, A and B. But in D, you can see that there's also some binding from the top and the bottom. And those 2 we're going to talk about a little bit more in detail. Okay. First, we're going to introduce globins as a family. If you remember from chapter 4 when we talked about protein structure, families are just proteins that are related by their structure and function. This is a pretty big family, very highly conserved tertiary structure. Highly alpha helical, connected by bends. And those bends are called the globin fold. Most of these globins function as oxygen transport or oxygen storage. There's 4 types of globins in humans and other mammals. We're only going to be talking about myoglobin, which is monomeric, so there's only one little protein, and it facilitates oxygen diffusion in the muscle tissue. And we're going to talk about hemoglobin, which is a tetramer, so there's 4 subunits, and it is responsible for transporting oxygen in the bloodstream. The other 2 globins we're not going to talk about, but you can see them there. You can look them up, look in the book at your own leisure, but we're not going to cover them in this class. First, we're going to talk about myoglobin. It's a monomer. It's a small protein, 153 residues. It also has 1 heme group. That 1 heme group has 1 binding site for oxygen. You'll hear terminology when related to the globin family, the proximal and the distal histidines. These are highly conserved. The proximal histidine, which I'll circle it here, is one that is close to that iron group. The distal histine is one that's located a little bit further away, but it still helps in coordinating that iron into the heme. Both of these are absolutely necessary for binding heme and for binding that oxygen. Before we go further in talking about things binding and becoming unbound, let's give a little bit of math to this. So we're going to talk about some equilibrium expressions for a second. Let's just use the generic protein and ligand. So protein is the P, ligand is the L. You bring those 2 things together, it's reversible. That's what these 2 arrows mean. They come together, you make a complex, the protein-ligand complex. Remember, there's no chemistry going on here. We just have something binding and unbinding. We can introduce a term called the association constant. Now, you're going to need to pay attention to capitalization here. The capital K with the subscript of A is the association constant. And it's equal to the product, which I know we're not doing an actual reaction, but we're making something new, which is the protein-ligand complex. And that goes on top of the equilibrium constant. And on the bottom, we've got the concentration of the protein and the concentration of the ligand. If we rewrite our little equation from the last slide, then what we're saying is that the forward reaction is the little k A. And the reverse is the little k D. So we're just establishing some terms. Mind your capitalization here. It's very important. The higher your capital K A is, the higher the affinity that the protein has for the ligand. If you have a ratio of Ka over Kd, that can tell you quantitatively whether or not the complex being formed is favored, or if it's more favorable for the protein and ligand to be separate. If you're setting up an experiment where you want to determine some of these different binding constants, if you have a ligand concentration that's really, really big in comparison to your protein concentration, so say that for your protein concentration, you're somewhere in the nanomolar range, okay? For your ligand, you might be in the micromolar to millimolar. Just going from nanomolar to micromolar, that's 1,000. That's 1,000 times, okay? So we're talking about really no change when all of that protein is bound to ligand. So when we say that the concentration of ligand is much greater than the number of binding sites, number of binding sites is like saying the amount of protein that you have, but sometimes you can have more than one binding site on a protein for a ligand. That's for another day. But what we're saying here is that if you have a really big concentration of ligand, the protein binding some of that ligand up is not going to change the concentration of the ligand. And that's something that's key to, again, designing experiments. And when we're working some problems, we're not necessarily going to be doing math per se, but we are going to be thinking through how to set up an experiment and, different concentrations of things and how to kind of evaluate a binding curve. So we covered the association constant. There's also a dissociation constant. So this is the capital K, lowercase d. Not to be confused with the lowercase k, lowercase d. I know, we could have been a little bit more original here, but usually constants like this have a k. So we're kind of stuck in a rut with semantics. I apologize for the whole field of biochemistry. Anywho, the dissociation constant is the reciprocal of the Ka. So that means that 1 over the capital K, lowercase a, is equal to the capital K, lowercase d. This is the equilibrium constant for the release of ligand. So for this, if you have a lower Kd, then that means that you have a higher affinity. Two ways to say the same thing. Now let's say that you've set up an experiment and you've got a really small amount of your protein, big amount of ligand. You're going through and you're doing your experiment. You're changing the amount of ligand. When you eventually plot all of your data, you're going to find a point where the concentration of L is equal to the capital Kd. And what that means is that half of the ligand binding sites are occupied. I'm going to show you what that looks like. So if you were doing this experimentally and you have your data, you're going to plot your concentration of ligand on the x-axis. It'll be something like micromolar or millimolar, something in that range. And the y-axis is going to be, well, it could be a number of things, honestly. It could be fluorescence, it could be absorbance, lots of things. But eventually you're going to get to a point where half of the binding sites are occupied. And you'll know what that is when you're doing this experimentally. You find that place on the y-axis. You go across to your binding curve, and then you follow that down to the x-axis. That is where you find your capital K, lowercase d, which is the same as 1 over your Ka. Here are some representative Kd values. Remember, this is the dissociation. So you want to have something that's really, really small. The smaller it is, the higher the affinity. And I really like this chart here because it gives you an idea of some of the different protein interactions that you might encounter and their relative affinities for ligand. As I said before, we're going to talk about enzymes in chapter 6. And enzymes actually have low affinity for their substrates. Think about why. You might see that as a question somewhere. So we talked about the generic protein ligand binding. Now we're going to apply that to myoglobin, which is what we started talking about in the first place. Remember, that is a protein that binds oxygen, and it is purely for the muscle. Okay, this is in muscle tissue. If you substitute in oxygen for ligand, then you get this equation here with oxygen over the sum of oxygen plus your Kd, the capital K, lowercase d. Same rules apply. So you can apply all the equation math that we did to this specific example for myoglobin. Again, you're not going to be responsible for these equations, but you will need to be able to look at a graph that is labeled properly and give me some information from it. Now we're dealing with oxygen. So a concentration of oxygen in some kind of aqueous solution, definitely hard to do. So in this case, we actually use partial pressure. That's a lot easier to measure. So same deal. The partial pressure of oxygen when half of your binding sites are occupied gives you your Kd. Believe it or not, carbon monoxide actually binds free heme more than 20,000 times better than oxygen does. I'll let you think about that for a second. Carbon monoxide binds free heme, not heme that's bound to myoglobin or hemoglobin, but free heme 20,000 times better than oxygen does. And it's really because of the differences in the orbital structures of these two molecules, oxygen gas versus carbon monoxide. So remember, in all the safety lectures that you may have had if you've done lab classes, or if you own a home, if your parents own a home, you're probably going to have a carbon monoxide detector in there. And the reason for that is carbon monoxide is a silent killer. It can replace the heme in your proteins, not as easily as with free heme, but it can replace the oxygen in hemoglobin and myoglobin. And if you have enough of it, if the levels in your home are too high, then it's actually fatal. So if you do not have a carbon monoxide detector, you need to get one. They usually have like a combination one that you can get that's a smoke detector and a carbon monoxide detector. Get it. Do yourself a favor. So, if carbon monoxide can bind heme way better than oxygen, what is it about myoglobin that actually helps it to bind oxygen a little bit better than it would natively, as in if it's just out in the wild, free heme? We come back to two things. One, this protoporphyrin group that has the iron coordinated in the center. And we also have the myoglobin protein itself. There are several interactions within this pocket that encloses the protoporphyrin and houses the oxygen when it is bound. And all of these interactions help to stabilize the bound state of the oxygen bound to this protoporphyrin. It coordinates with the iron. So what you can see here is that we've got the distal histidine, excuse me. It is hydrogen bonding with this oxygen. And the oxygen is in turn now coordinating with the iron. So these interactions help to make oxygen a much better binder to the heme group. That 20,000-fold stronger binding affinity, it becomes 40-fold. Big difference. Carbon monoxide still binds significantly better, but 40-fold better is something we can deal with. Much better than 20,000-fold. So proteins and all these interactions really make a huge difference in how our bodies and our cells operate. Now we're going to move on to hemoglobin. Hemoglobin transports oxygen. They travel around in your red blood cells, the erythrocytes. And that is really their main function, is to carry around hemoglobin. Your arterial blood is approximately 96% saturated with oxygen. Peripheral blood, so everything, outside of that, about 64% saturated. Hemoglobin is a tetramer, which means that there are 4 individual little monomers that come together to make hemoglobin. So when you see something called a tetramer, or it is a tetrameric protein, that speaks to the quaternary structure. And along with each of those monomers, they each bind their own heme group. So hemoglobin has 4 heme groups, which means 4 possible places for oxygen to bind. Adult hemoglobin, which there is a fetal hemoglobin that we're not going to talk about. Adult hemoglobin has 2 globin types. There's 2 A chains and 2 B chains. This is a comparison of myoglobin and the beta subunit of hemoglobin. You can see that they're very similar. Now, in terms of primary sequence, there's very low sequence similarity between these two. However, the structural similarity, which you can see in the figure on the right, pretty high. Their heme groups are pretty much in the same place. All of the alpha helices and the loops are pretty much in the same place. Loop regions tend to be more flexible, so those can kind of move around a bit. But the helices, by and large, are in the same place. There's strong interactions between alpha and beta subunits of hemoglobin. So all those tiny interactions that individually don't mean a whole lot, but when you bring them all together mean the world: hydrophobic effect, hydrogen bonding, and salt bridges. Those three things are key to keeping hemoglobin together. So if you look at the A1 or the alpha 1 beta 1 and the alpha 2 beta 2 interfaces, there are more than 30 residues involved in all of these different types of interactions. And alpha 1 beta 2 and alpha 2 and beta 1, there's 19 residues. So again, this just illustrates the point. You need a lot of these tiny little interactions to come together and make a big effect of keeping this monster protein together. Hemoglobin actually undergoes a structural change when oxygen binds. There's the T state and the R state. The R state is the one that actually has a higher affinity for oxygen. I think about it this way. R state is the ready state. R equals ready. The T state is actually more stable when oxygen is absent. So deoxyhemoglobin, deoxy means no oxygen. We're going to be looking at these two states in a little bit more detail, and we'll see the changes that happen when you transition from the T state to the R state. But in a nutshell, the ion pairs change between the different alpha and beta subunits. So the T state has a certain set of interactions that allows it to stay together. The R state has a slightly different set of interactions that stabilizes its conformation, including the oxygen being bound. The T state has a greater number of ion pairs, and that's at the alpha 1 beta 2 and the alpha 2 beta 1. You don't need to remember specifically where it is, but just remember that the T state has more ion pairs than the R state does. Some of the key interactions I'm going to highlight here. There's a lysine that is hydrogen bonding with the backbone of this histidine on the beta 2. So this is the lysine R group, and this is from the backbone of a histidine residue on the beta subunit. So there's one hydrogen bond there. We also have some hydrogen bonding between an asparagine on a loop between two helices and a histidine on that same beta subunit. So those are two of the key interactions between the alpha and beta subunits that help stabilize that T state. When oxygen binds the T state, which remember, reduced affinity is different from no affinity. Oxygen can still bind the T state. It's just, it doesn't as readily occur. Once it does, it triggers a conformational change to the R state. So the alpha and beta subunits kind of twist and rotate. And there's a pocket between the beta subunits that gets more narrow. And some of those ion pairs are broken and new ones form. We'll show you that in just a second. So this is the T state on the left, the R state on the right. Remember though that histidine that was hydrogen bonding with the asparagine. Once that twist happens with the bound oxygen, we can't have that interaction anymore. And now that histidine is looking at its brother histidine on the other side. So this is the histidine from the beta 1 and the histidine from the beta 2. They're no longer in that hydrogen bond with the asparagine. There's also some conformation changes near the heme group itself. In the T state, this is what it looks like, okay? We've got helix F, which has a lot of the residues that help coordinate the heme group. We've got our proximal histidine. It actually gets closer in the R state to that iron. We also have some changes with the valine and leucine above that histidine that they kind of twist and shift a little bit to actually get closer together. And they have some van der Waals interactions here. So we've got van der Waals there. Let's get a different color. We've got the proximal histidine gets closer. And then we also have a change in that leucine residue as well. The leucine kind of turns and starts interacting more to stabilize that heme group, that protoporphyrin. So that's again more kind of van der Waals. If I sound excited, it's because I really like this stuff. This is my jam right here. I love looking at how proteins interact with their ligands. So excuse me if I get a little bit excited. Maybe I talk a little bit faster. I apologize. It's posted on YouTube for a reason. You could pause it, rewind it back as many times as you want to. Like, man, she's so excited. She talking too fast. I'm sorry, guys. This is what I like. So we're still talking about hemoglobin. Lots of changes that happen to the structure of hemoglobin in the presence of oxygen. Because of that, we can also see some interesting things when we look at the binding of oxygen via a binding curve. It turns out that oxygen binds hemoglobin cooperatively. Now, in your textbook, there's some talk about Hill coefficients that I did not include in here. We're not going to talk about Hill coefficients. I just want you to know that hemoglobin binds oxygen cooperatively. Jumping into all of the math and understanding Hill coefficients and how you can use those plots to determine whether or not something is binding cooperatively is a bit above our pay grade. I'm not going to do that. If we were doing maybe like a 400 or a 500 level course, we'd absolutely include it. But for now, if it's something that interests you, if you like graphs and math, by all means read it. Come to me and ask questions. But if you don't like math, you don't have to worry about it. So we're looking at two different regions here because we've got two different regions of the body to think about oxygen. We have to think about the oxygen in tissues and the oxygen in the lungs. Now, the oxygen in the lungs, the partial pressure for oxygen is going to be much higher in the lungs than in tissues. So we can look at hemoglobin when it's in that low affinity state. So all of your monomers are in the T state. Then bam, you've got an oxygen binding to one of those heme groups. And that triggers the change from that T state to the R state. That means that the rest of the monomers in there that make up that tetramer are going to start changing to the R state. They don't have to have an oxygen bound to change to the R state. All that needs to happen is an oxygen binds to that T state that stabilizes the R conformation that encourages all the others to switch to R. Proteins are dynamic, so they're constantly kind of shifting and moving, and they are already transitioning back and forth between T and R states. But once an oxygen binds, it encourages the R state to be present more readily. So in the middle here, this curve is transitioning from the low to high. You've got a mixture of T and R within the same complex of the hemoglobin. Then eventually, you have all R. And that's where you have the greatest affinity for oxygen. So if you were to look at each of these curves and try to figure out the KD, this is how you would do it. Now for the high affinity one, it's such a high affinity that KD is super teeny tiny. Remember that the KD, capital K, lowercase d, is the dissociation constant. And it is low for high affinity interactions. When you have all of your hemoglobin in the R state, then it's really high affinity, and that KD is going to be really low. It looks like that curve is hugging that y-axis so tightly that with all three of these binding curves on here, we can't see anything. I also want to point out that the partial pressure of oxygen in the tissues is lower than in the lungs. Think about that and why that should make sense to you. We'll talk about some of these questions in class, but I'm going to try to kind of give you clues as to things that could show up on quizzes or exams that aren't explicitly stated in the lecture, but you have enough information to form an educated response. Because we're talking about hemoglobin, and it's a really important protein because it transports oxygen throughout your body, things that are really important like that usually have tons of regulation associated with it. One way that you can regulate a protein is by introducing some kind of allosteric interaction. So let's define some terms. Allosteric proteins, like hemoglobin, they have a binding site for a ligand that affects the binding properties of another site on the same protein. So we know about the oxygen binding site with the heme, but there's another binding site on hemoglobin where other modulators can bind and control the binding of oxygen. More terms. Modulators. A ligand that binds to an allosteric protein causes some kind of conformational change. Homotropic means that it's the normal ligand modulator pair. So if you think about our hemoglobin example, it would be hemoglobin and oxygen, right? Heterotropic is the modulator other than the normal ligand. So something else that hemoglobin normally wouldn't bind, that it doesn't do anything in terms of transporting it, but there's a site for a molecule to bind to control the other binding site that's far off somewhere else. So let's talk about it. Just as a reminder, remember that carbon monoxide has a super high affinity for myoglobin, also has one for hemoglobin. And for hemoglobin, it's 250-fold greater than oxygen. So again, if you do not have a carbon monoxide detector, you should get one. They have the ones where it's a combo, for the smoke detector. But honestly, I recommend having one that you can plug in an outlet that's lower down, because carbon monoxide is a little bit more on the dense side in terms of gases. So if you're going to have a higher concentration, it's probably going to be lower to the ground. Again, stay safe. Lots of things that you learn when you own a home or if your parents own a home. So keep yourself safe. Hemoglobin can also transport hydrogen and carbon dioxide. Those are two products of respiration. We're going to talk about respiration a lot when we get into metabolism. Carbonic anhydrase is an enzyme that catalyzes the hydration of carbon dioxide to bicarbonate. Now you might be scratching your head like, bicarbonate. I feel like I've seen that before. We're talking about red blood cells, bicarbonate, blood. Yes. Remember that bicarbonate is part of the buffer system in your blood. So that is the conjugate base that helps to regulate the pH and keep it somewhere in the range of 7.3 to 7.4, somewhere in there. Carbonic anhydrase, ironically enough, my thesis advisor, when she was a graduate student, worked on carbonic anhydrase and characterized its enzymatic activity. So why do we care? Well, we need to talk about something called the Bohr effect. The structural effects of the hydrogen molecules, hydrogen atoms, the H+, and the carbon dioxide binding to hemoglobin actually favors the T state. Now, thankfully, these two things bind reversibly. And so if you have oxygen bound, you're not going to have hydrogen and carbon dioxide bound. So they are inversely related. The Bohr effect describes the effect of pH and concentration of carbon dioxide on the binding and release of oxygen by hemoglobin. So there's this equation here where we have a hydrogen added on to the hemoglobin. So that HHB plus, that's the added hydrogen. When there's oxygen in the mix and it binds, you release that hydrogen and instead you have oxygen bound. When you have an increase in hydrogen, what does that mean for your pH? If you increase the concentration of H+, then that leads to a decrease in your pH. More acidic. So when oxygen concentrations are high, hemoglobin will bind oxygen and it will release hydrogen. When the concentration is low, and remember you can think about concentration or partial pressure, same deal here for oxygen, then hemoglobin is going to release oxygen and bind hydrogen. Thinking about the goal of hemoglobin in transporting oxygen to all the tissues, this should make sense. You don't want hemoglobin holding on to oxygen forever, because then it won't actually transport it anywhere. It will just store oxygen. That's not what we're going for. We're going for something that can pick up oxygen from a highly oxygenated place and deliver it to a place that has low oxygen, and then keep it pushing, keep cycling it over and over again. So look at this slide. Think about how the pH of the lung versus other tissues might play into this, just giving you some hints on things to think about for how to prepare for the exam. Something else that we can talk about in terms of modulating the binding activity of hemoglobin is 2,3-bisphosphoglycerate. That is a bit of a mouthful. Usually it's abbreviated BPG. This is an example of a heterotropic allosteric modulator. It binds to a site that is far away from the oxygen binding site, and it reduces the affinity of hemoglobin for oxygen. BPG is something you're going to see again when we cover glycolysis. It is actually one of the metabolites in the process of glycolysis. So this is just a reminder that all of the processes that your body is doing, they're related. It's kind of like one of those, everyone is related by like 6 degrees. Everybody knows somebody within 6 degrees of another person. Like you can relate each person. That's how it is with your body. All of these molecules that are being produced, maybe they're waste, maybe they're, building blocks, maybe they're modulators. All of these chemicals have roles in maintaining your body and keeping it healthy and strong. So there's a lot to learn here in terms of metabolism and modulation of binding activities, modulation of enzymatic activity, which we'll cover in chapter 6. So this is not the end-all be-all. We're not just going to talk about hemoglobin in our class, but we'll look at other examples, and you'll be able to apply the knowledge that you have to those other examples to glean information and talk about them intelligently. So if we look at hemoglobin, we've got all of our heme groups. And where is that BPG? Right here in the cavity between the beta subunits, and it prefers to bind in the T state. That cavity is lined with lots of positively charged residues, and BPG has a lot of electronegative oxygens. When it's bound, it stabilizes the T state, which means that you're going to have reduced binding of oxygen. So we finished our walk through the globins. We talked about myoglobin and hemoglobin. Spent considerably more time on hemoglobin because, well, it's bigger and there's lots more to talk about. Now we're going to talk about another example of a protein ligand combination. We're going to head on over to the immune system. This is going to be a brief overview of section 5.2. There's a lot more in here about the immune system, but this is not an immunology class. We're not talking about the immune system. So if you're interested in that, by all means, read the whole section, but you're not going to be responsible for any more than what is in this presentation. In general, when we're talking about an immune response, we're talking about a highly coordinated set of interactions among several types of proteins, molecules, and cell types. So it's a big deal to mount an immune response. And the goal of your body is to distinguish the molecular self from the non-self and destroy all of the things that aren't you. So this gets rid of viruses, bacteria, other pathogens, molecules, all the things that are not you. And your body has all types of proteins and cell types to identify you versus not you. One of the major classes is antibodies, and a major class of antibodies is immunoglobulin G, or IgG. It's one of the most abundant ones in the blood. It's got 4 polypeptide chains, 2 heavy and 2 light chains. You're actually able to cleave IgG with a protease named papain, and you release a couple of different fragments that each have an antigen binding site. We're going to talk about what an antigen binding site is in a minute. The constant domain is the one that has the immunoglobulin fold. So all immunoglobulins have the same structural motif that classifies them as being part of the immunoglobulin fold or family. So this is a cartoon of what IgG looks like. Up top, you have your antigen binding sites. So there's one here and one here. And each of those sites is unique. That allows you to recognize a whole host of different antigens. They form a really tight complex with an antigen. So it's a pretty high affinity interaction there. This is another way to show the structure of IgG. So we've got another cartoon on the left, and on the right we have a crystal structure. As practice, you should look at this crystal structure and try to identify some of the secondary structure that you see. Again, these are the antigen binding sites. This bottom part is the constant domain. And as part of the antigen binding site, you have that's the variable domain. So that's where that antigen binding site is housed. You don't need to remember the structure per se outside of knowing where the antigen binding site is and the fact that that is what gives all these different immunoglobulins their ability to recognize different antigens. Antibodies bind really tightly and really specifically to their antigen. And there's what's called induced fit, which means that you have, in this case, an antigen that binds and then causes a shift and change in what the protein looks like after it's bound. So there's binding, and then there's a shift in the protein and sometimes even the ligand that causes even tighter binding to occur. You can have really, really low KD values, as low as 10 to the minus 10 molar. If you look closely at these space-filling models of the conformation of this antibody without an antigen and then the one with an antigen, you can see some differences. You don't need to know these differences, but I'm just pointing them out so that you can see that there are some kind of major shifts in what this protein looks like. You can see that kind of mountain poking out there. That has flattened out quite a bit. So that's one change that you can see between no antigen and antigen bound. If you also look at this blue part over here, it has shifted quite a bit. Those are just two examples of how the interactions within the protein shift when the antigen is bound. These shifts encourage tighter binding of the antigen. There's two types of antibody preparations that you can use in the lab. There's polyclonal and monoclonal. Polyclonal you produce by injecting a protein into an animal, or it could be a piece of a protein that you're interested in developing antibodies for, and then you collect a mixture of antibodies that recognize all different parts of the protein. You can also get monoclonal antibodies by synthesizing a population of identical B cells, and then they will produce something that's monoclonal antibodies. They're all homogeneous. They recognize exactly the same thing. These can be really helpful in a lab setting. Western blots use antibodies to detect a protein. You'll also see Westerns called immunoblots. So immunoblot, Western blot, Western, all the same thing. And pretty much what you do is you run a protein gel that has your protein of interest on it, or you think it has it on it. You should have some kind of a control. So you use an SDS page gel, has your protein within that matrix. Then you coat the surface with a blocking solution, and that's just to make sure that there's no non-specific protein interactions between the antibody that you're about to add and all the protein that's on your gel. You add your primary antibody, which has specificity against your antigen, so the thing that you're interested in. Then you add a secondary antibody that can detect the primary antibody. So if we're following along in this picture, we've got our primary antibody, and then the secondary antibody recognizes it, the primary. On that secondary antibody, there is either an enzyme that can react with the substrate to cause a color change or a change in fluorescence or something like that you can detect. They also have secondary antibodies that, or actually they have primary antibodies that have a fluorescent tag attached to them. So you don't even have to use a secondary antibody all the time. But the bottom line is you have to have some way of detecting that you have bound primary antibody. And when you do that, you can go from having a protein gel that has lots of proteins to only visualizing your protein of interest. Protein of interest. I usually abbreviate that POI. That doesn't mean that other proteins aren't present, but it means that the only ones you see are the ones that you care about. It's a very powerful technique for protein detection. That's it for Chapter 5. Thanks for watching. Make sure that you come to class for all the practice problems we're going to do, because you'll get to practice asking some of the same types of questions of different protein ligand pairs, and you'll learn more about what I'm expecting you to be able to do on an exam or in a quiz. So until I see you in class, stay safe.